There are many differences between prokaryotic and eukaryotic cells, but one of the most obvious is the presence of organelles. Though there is only one membrane that surrounds the entire cytoplasm, there are many smaller lipid bilayers within the cell that obstruct the free exchange of cellular metabolites. The degree to which a given compartment is permeable to a species is not simple, and there are many active transport processes associated with specific organelles. Thus, the degree to which a biomolecule is isolated within an organelle is not obvious. Mitochondria are the ATP-generating organelles of a eukaryote. They do encode their own genome and encode some of their own proteins. The degree to which the mitochondrial genes are encoded in the organelle versus in the nuclear genome differs between different organisms. In all cases, it is technically challenging to modify the DNA of a mitochondrion. So proteins intended for mitochondria are usually sent there by use of a localization signal. The mitochondrial targeting peptide is a helix with a few hydrophobic amino acids and positively charged amino acids. This signal is recognized post-translationally, so a protein will first exist as a cytoplasmic intermediate before being imported into the organelle. Secretion in eukaryotes is mediated by a compartment called the endoplasmic reticulum, or ER. The process of ER secretion is highly analogous both in sequence and effect as E. coli sex secretion. Both proteins that will be secreted to the external environment, as well as those being targeted to the membrane for surface display, will go through the ER. The ER is the starting point for both integral and associated proteins and most glycosylated proteins. The signal for ER targeting is a 5 to 10 hydrophobic peptide on the N-terminus of the protein. As nascent protein chains emerge from the ribosome, as shown in this illustration, these peptides are bound by signal recognition particle and recruited to the ER. Translation then proceeds coincident with transport across the ER membrane. Unless it is a membrane protein, the newly released peptide is cleaved off and free within the organelle. Additional signals can be placed into a protein coding sequence to target it to other places in the cell. For example, a DXE motif in the C-terminus of the protein is the signal for targeting to the Golgi via COP2 vesicles. The nucleus compartmentalizes the genomic DNA, and thereby necessarily the processes of replication and transcription. The protein components of the nucleus, however, must be translated in the cytoplasm like any other protein and then retransported into the nucleus. The nuclear localization signals are highly positively charged peptides. The sequence PKKRKV from the SV40 large T antigen is one commonly used sequence. Another one from nucleoplasmid is a second option. The nuclear localization signal, or NLS, is usually internal to a peptide sequence rather than at the ends, but it can be anywhere. Like with mitochondrial targeting, nuclear localization is post-translational. Nuclear localization is necessary for access to the genomic DNA, so any engineered behaviors such as gene delivery methods necessarily involve NLS signals. Peroxisomes or organelles usually used to degrade long-chain fatty acids. It is a highly oxidizing environment. There are two types of signals that can transport a protein to the peroxisome. PTS1 signal is a C-terminal SKL peptide. PTS2 signal is an N-terminal sequence. The peroxisome is being explored as an engineering platform today. Lysosomes degrade proteins and other debris in the cell. Targeting proteins to the lysosome involves a C-terminal signal called the dileucine sorting signal. Though you can send a protein to the lysosome, this usually results in its destruction. The pH is 4.5, making this chemically denaturing for most proteins. Thus, the targeting proteins to the lysosome is usually a control strategy for intentionally removing a protein from the cell or causing a protein to recycle in the cell that otherwise would accumulate. Finally, we have the chloroplasts and related plastids. These are the photosynthetic centers of plants and other single-celled eukaryotes such as Volvox, Euglena, or Chlamydomonas. The chloroplast is the organelle most similar to a prokaryotic cell with an extensive genome. Nevertheless, you would still usually genetically modify the nuclear genome first rather than the chloroplast genome, simply because it's easier to do. There is a large family of related organelles called plastids in plants. We'll come back to these in a later chapter on plants. Other processes such as starch production happen in these organelles. This family of compartments are also the specialty workshops of plant cells where interesting natural products get produced.